Next, I will talk about the RNA-seq rate distribution and the differential expression analysis. Here, again, I will show you an example with gene A and with 3 replicates in each condition to compare 2 conditions. The 2 groups can be, for example, normal versus tumor, or control versus drug treatment, or developmental stage 1 versus developmental stage 2. So anyway, in group 1, for gene A, we have, this is the raw read count, and this 22 million or 30 million or 18 million is the total read, mapped read. And in group 2, similar, 45, 55, and 31 is the number of raw read count for gene A, and the second number, 20 million, 28 million, and 27 million are the total read, mapped read in the library. So to determine if gene A is differentially expressed, we should examine whether the proportion of reads aligned to gene A tend to be different for the samples in group 1 comparing to the 3 samples in group 2. And here we have to think about it. Should we normalize for gene length and the library size when we do such comparison? So if we compare the same gene, then very likely the gene lengths are consistent across the 2 conditions. So there is not necessary to compare to gene length. But other than that, there is a more important factor we have to think about, that is statistical power. So for longer gene, that with more reads aligned to this gene, statistically speaking, is more powerful than the short genes with less read aligned to it. Similar to the library size, if we sequence deep and we have tons of reads, then statistically it's more powerful to say that to distinguish whether this gene is differentially expressed or not, comparing to the shallow sequenced library size. So this is important. This is why we use raw read count or count per million in the differential expression analysis, and let the differential expression analysis tool to do the normalization, to do the proper normalization. And we do not use FPKM or TPM. In terms of RNA-seq read distribution, read count distribution, let's think of some simple cases. There are some rare events. For example, the number of patients arriving in an emergency room between 10 to 11 p.m., or the number of phone calls received at a call center within 1 hour. Do you know what distribution we use to model those sparse events? Yes, Poisson distribution. Similarly, the number of reads mapped to a gene of 1 kb long, for example, over a 3 billion base pair genome. This is also a sparse event. It also uses Poisson distribution to model such sparse event. So let's recall that in Poisson distribution, the mean of the Poisson distribution is the same as the variance of the Poisson distribution. So if we plot our data, RNA-seq recount data, if we plot the x-axis as the mean count, and the y-axis as the count variance, then we can assess whether the Poisson mean variance relationship holds or not. So let's look at the plot in this paper. This is based on fly RNA-seq data. The mean is in x-axis, it's in log scale. The variance is also in log scale, it's in the y-axis. The purple line is the variance mean relationship modeled by Poisson model. The orange solid line is the mean variance relationship modeled in RNAseq software, and the orange dashed line is the relationship modeled in HR software. We can see that both the orange line, the dashed orange line, and the solid orange line fits the data better than the purple line. So a number of softwares are developed to account for the overdispersion in RNA-seq. The overdispersion means for the Poisson distribution, the mean and the variance are same, but for actual data, the variance is actually higher comparing to the mean. So it's generally accepted that negative binomial-based methods performs better than Poisson-based counterparts, and HR and the RNAseq and the RNAseq are both based on the negative binomial distribution, and these two softwares are among the best performers and the most widely used software. And these two software, the difference is they implement different strategies for this dispersion estimation. So let n be the total number of RNA-seq reads, and the theta be the relative abundance of a gene. Then the read count can be modeled as the binomial distribution n and theta. Once the number of reads is large, the distribution of the distribution of the negative binomial can be approximated by the Poisson distribution. To model the count for group I, gene J, and sample K, we can model it as this Poisson, or we can model it as this negative binomial distribution. So how to select a software? First, this is just a general guide. First, you need to know what expression level you want to compare. If it's exon level, or if it's isoform level, or if it's a gene level. If it's gene level, then what experimental design? Is that a multiple factor or just a one factor? If it's just a one factor, then how many replicates? Whether it's just less than 5 replicates, then you can use RNAseq2 or HR or Limavu. And if you have a lot of replicates, you can consider other method. And in principle, if you have large enough replicates, you can just use t-test.